Hello everyone. Now in this current video of mine, I will be starting up with the chemistry of carbohydrates. Monosaccharides chemistry we have finished in the previous video. Now in this video we will be starting up with the disaccharides, oligosaccharides and polysaccharides. Now we all know that if we talk about disaccharides, it is clear from the word itself, di means two. That means when one monosaccharide will combine with the second monosaccharide, simply it is going to lose a molecule of water. And the bond so produced will be known as a glycosidic bond, which is very important. So always remember, one member of carbohydrate combines with another member of carbohydrate, losing a molecule of water, forming a glycosidic bond, which is commonly known as COC, or I will simply write glycosidic. So that is what exactly happens. So disaccharides are the ones which are formed by the condensation of two monosaccharide molecules. And if we talk about the physiologically important disaccharides, there are physiologically important. The first one is lactose. Now, lactose, we all know it is known as milk sugar. And if we talk about the components present in lactose are galactose and glucose. And if we talk about the linkage between them is beta 1,4. But it comes by default that it has to be beta 1,4 glycosidic linkage, because this linkage I have already mentioned here, right? So this is one very important disaccharide. Second is maltose. Maltose, which is also known as malt sugar. Malt sugar. And if we talk about the components, it has glucose plus glucose, and both are in alpha form. So the linkage is alpha 1,4. Then the third one is sucrose. We call sucrose as the table sugar or the cane sugar, we can say. And sucrose has glucose and fructose. But the linkage here is alpha 1, beta 2. Right. Then we come over to the fourth one, trehalose. Trehalose is present in the lower animals, and it has glucose plus glucose, which is linked as alpha 1,1. And then the fifth one is lactulose. And basically this lactulose is a synthetic sugar. Synthetic, and obviously if we talk about its function, it actually disturbs the osmolality of the body, and it can be used as a laxative also. And if we talk about the components, it has galactose plus fructose. And very important, galactose is alpha and fructose is in beta, but alpha 1, beta 4. So this is the table which you have to remember in disaccharides. So I can simply just give the headings. This is the name of the sugar, the common name. These are the components. And this is the linkage which is present. Now another important feature here which you have to remember is lactose and maltose are reducing sugars. And if you want to know again the detail about reducing sugars, you can refer to the previous video, the first video on carbohydrate chemistry. If we talk about sucrose and trehalose, they are non-reducing sugars. And lactulose itself is a laxative which will not come under either reducing or non-reducing sugars. So always remember, whenever we talk about lactose, maltose, they are reducing sugars, and trehalose and sucrose are non-reducing sugars. And another important point about this is this sucrose is also known as invert sugar. Sucrose, invert sugar. Why? What is the reason behind that? This is again very important question. Now the reason behind this is that sucrose, if we just look at the structure of sucrose, it appears to be dextrorotatory because it is formed of on hydrolysis. It is going to give glucose and fructose. Okay. So, if we look at the structure of sucrose, it appears to be dextrorotatory. But if we measure the angle of rotation of glucose and fructose, fructose overweighs in that because glucose has plus 52.5 degrees, whereas fructose has minus 91 degrees. So, simply I can say there is an inversion of configuration. So, what is the reason? Appears to be dextrorotatory, but due to the more levorotatory power of fructose, as you can see the angle of rotation, it is minus 91, much more than plus 52.5 degrees. So, but due to more levorotatory power of fructose, there is inversion of configuration. Inversion of configuration. And that is why it is known as invert sugar. And the enzyme required to degrade will be invertase. So, the most appropriate terminology to degrade the enzyme for sucrose is invertase. Then, what is the clinical correlation of this? The most important thing which you have to know about disaccharides is lactose intolerance. Now, if we talk about lactose intolerance, it is clear from the word itself. The person is intolerant to lactose because we have seen that there are so many people, whenever they digest, whenever they try to have those dairy products, whether it is milk, whether it is anything related to dairy. So, they will start having that bloating kind of thing. Why? Because they are intolerant to lactose. So, that is why it is known as lactose intolerance. And what is the reason behind that? The reason behind that is simply absence or deficiency of lactase. Right? And what are the simple, I can say that cannot digest dairy products. Right. And what are the clinical features? The most important clinical feature will be it will lead to bloating in the stomach, which will lead to flatulence in the stomach, and which might lead to vomiting or might result into diarrhea. Right. And what is the treatment? The treatment is simple. The patient who is suffering from lactose intolerance, he should actually take skimmed milk powder, because skimmed milk powder there are less chances of this because it has a little bit of fat content also. And second important thing is he can switch over to lactate pills where this enzyme is already there. So, treatment is, you can simply say lactate pills, which are nothing but the enzyme, and second is skimmed milk powder. So, these are the two very important properties which you have to remember about disaccharides. One is the difference between reducing sugars and non-reducing sugars. Just now I told you, and वैसे भी you just have to learn it by heart, that all sugars are reducing except sucrose and trehalose. And second important thing is lactose intolerance, absence or deficiency of lactase enzyme. So the person will be unable to digest the dairy products. Then, since we are discussing about reducing sugars, the most important thing which we have to remember here is there is one test which distinguishes between reducing and non-reducing sugars. And that test is Benedict's test. Benedict's test we are going to again discuss when we are going to study about the qualitative test on carbohydrates. There again the concept of reducing and non-reducing will come, right? So this is all about disaccharides. Then the third class is oligosaccharides. Now if we talk about oligosaccharides, simply I can say that they consist of 3 to 10 monosaccharide units. But always keep one thing in mind, again the bond will be glycosidic. So they all have glycosidic bond. Now, if we talk about the function of oligosaccharides, they play a very important role. Why? Because they are actually present on the surface of the cell membrane, and the most important function of oligosaccharides is, since they are present on the surface of the cell membrane, cell to cell recognition. Since they can recognize different cells approaching the cell membrane, so they have an ability to distinguishing between self and non-self. So distinguish between self and non-self, which becomes the basis of immunity. Otherwise it might lead to autoimmune disorders. So these two functions in fact covers all the properties of these oligosaccharides. Now coming over to these oligosaccharides, because we know whenever we I would say study the structure of cell membrane, carbohydrates are there, proteins are there, lipids are there, there are other complex aggregates also, like glycoproteins are there, proteoglycans are there. So basically the main funda behind that is to put a negative charge on the cell membrane. Now why the cell membrane has to be negatively charged? Because it is selectively permeable. It won't allow all the molecules to pass through it. So it will allow only selective molecules to pass through it, and this is only possible when we are putting a charge on that. So just keep this thing in mind. So I can say that these oligosaccharides, they have a tendency to form two types of substances, proteoglycans and glycoproteins. Now what is the difference? It is clear from the word itself. Whatever is present as a suffix will always be more. So if I talk about proteoglycans mein glycan means carbs or mainly GAGs are 95% and proteins 5%. On the other hand, glycoproteins mein proteins are 95% and glyco means carbs are only 5%. So depending upon this composition, the solubility will also vary. Now the question is whether they are proteoglycans, whether they are glycoproteins. If I have to draw the structure, I can simply say that it is like this, like this. It's a mesh of a structure, just like, bottle brush flower, just like that, like this. Okay, now whether they are glycoproteins, whether they are proteoglycans, just keep one thing in mind, they have two types of glycosidic bonds. One is O-glycosidic bond and the second one is N-glycosidic bond. Now what is the difference between O and N? If we talk about O, I can simply say that a carb is connected to a protein. Proteins as such, they don't have any independent existence. Proteins which are formed of these amino acids. Now, if O-glycosidic bond is there, that means these amino acids will be having either serine or threonine, because serine and threonine are hydroxyl group containing amino acids. And again, if we talk about serine and threonine, we know that serine protease is a very important group of enzymes which is present on the surface of enzymes, which helps in actually accelerating the entire carbohydrate metabolism also. If we see the structure of glycogen synthase, glycogen phosphorylase, it has serine residues, OH. So this is O-glycosidic bond. If we talk about N-glycosidic bond, obviously the carbs which are attached to the protein or these amino acids which are having amino group in them. जैसे lysine is there, arginine is there. So it is NH2 containing. Amino acids. So these are the oligosaccharides. Very important to remember. Stachyose is there, raffinose is there. They will come under this category. Then we come over to the last class of carbohydrates, polysaccharides. Now, as the word suggests, poly means many monosaccharides combined. So, it is having more than 10 monosaccharides. And if we talk about the detailed classification of these polysaccharides, they are classified into two subclasses. One class is homopolysaccharides and second is heteropolysaccharides. Now, homo means identical units, and if we talk about hetero, non-identical units, right? So, these homopolysaccharides are further divided into two subclasses. One category, glucosans, and the second category, fructosans. And if we talk about heteropolysaccharides have basically two components. वैसे तो it is divided into acidic, basic, neutral, but here we don't go into that much detail. We just have to go superficially that heteropolysaccharides have two basic components, amino sugars and uronic acid. So, if we talk about amino sugars, it can be simply, you can say, galactosamine, or it can be glucosamine. And if we talk about uronic acid, it can have glucouronic acid or it can have idouronic acid. So, depending upon their, you can say the synthesis, depending upon their presence, they are having different components. And if we talk about glucosans ka the most common example is starch, glycogen, dextrans, dextrans and cellulose. And if we talk about fructosans mein there is only one example, inulins. And it is clear from the word itself, homo means identical. So, glucosans only glucose, fructosans only fructose. So, this is the brief classification of the polysaccharides. Now, all are important. So, first of all, we are going to discuss the homopolysaccharides, under which we are going to discuss starch. Now, if we talk about starch, simply we know that it is the reserve food of plants. Food of plants. And if we talk about starch, it has two forms. And what are the two forms? It has one form which is amylose, which is actually linear. So if it is linear, it will have only one linkage, which will be alpha 1,4. And the second one is amylopectin. The basic difference between amylose and amylopectin is amylopectin is a branched structure. Branch structure means it has alpha 1,4 to hoga hi hoga. Along with this, it will have alpha 1,6 linkage, which is very important, which is giving a branched kind of structure to starch. Okay, so depending upon again you can say the structure, we can explain the solubility. So more the linkage, lesser will be the solubility. It is but obvious. So if we talk about the solubility, amylopectin is insoluble, or you can say least soluble, and amylose is slightly soluble, because whenever we have to mix the starch, we always need to warm it little bit. So you can say that amylopectin form of starch is insoluble, whereas amylose is soluble. Then insoluble, amylose, amylopectin, alpha 1,6, alpha 1,4. Then always remember, starch gives blue color with iodine. This is one of the tests. I can simply say that starch gives blue color with iodine. Right? And this coloration, whenever we, basically the funda behind this is, this iodine is going to fix itself into the glycosidic linkage. The moment we heat the solution, iodine will go out, coloration will disappear. If it is allowed to cool down, the coloration will again appear. So this becomes a confirmatory test also to check starch. So I can say blue color with iodine, coloration disappears on heating and coloration reappears on cooling. Very important. So this is starch which is existing in amylose and amylopectin. And another important thing is, starch always has one reducing end. It will have, because somewhere down the line it will have one carbon atom, carbon 1 free, because the chain is going to demolish at one corner. So it has one reducing end and one non-reducing. So this is all about starch. Then second one is glycogen. Now starch, we know that it is the reserve food in plants. Glycogen is the reserve food in animals, reserve food in humans. Let's be more precise, reserve food in humans. We all know that glycogen is synthesized in the liver as well as the muscle. So it is synthesized in the liver and muscle by a process known as glycogenesis. And whenever the body requires energy, this glycogen will be broken down. So I can say synthesized and broken down by glycogenolysis. So these are the two very important terminologies which obviously we are going to discuss under glycogen metabolism in detail. But here, since we are discussing the chemistry, so what is the structure of this? So if we talk about the structure of glycogen, the most important difference, actually obviously it has to be branched. That means if it is branched, it has alpha 1,4 linkage as well as alpha 1,6 linkage. But what is the difference between the structures of starch and glycogen? The most important difference is that branching appears after every 10th to 12th residue. After every 10th to 12th residue. Or in a nutshell, I can say that it is more extensively branched or more intricately branched. Very important, right? So this is the main difference between starch and glycogen, that it has to be more intricately or extensively branched because it is synthesized in the body. And it is said that after 10 to 12 hours of fasting, the entire glycogen reserves will be depleted to form glucose. Okay, so this is one very important aspect here. So if I have to say that, suppose if the structure of starch is like this, alpha 1,4 and alpha 1,6, alpha 1,6. The structure of glycogen would be alpha 1,4 and alpha 1,6 like this. So this is the linear one and this is the branched one. And this is the difference. And another practical difference is this: is starch gives blue color with iodine, glycogen gives a red color with iodine. Right. So these are the two very important glucosans. As you can see, they are formed only of glucose. Then after this, then we come over to the third one, dextrans. Under dextrans, we have to just remember one thing: they are formed of glucose only, and simply you can say they are the bigger units of maltose, to be very precise and simple. Bigger units of maltose. And always remember, maltose, we know it's a disaccharide. Dextrans are there, then one more molecule, limit dextrans are there. But both are produced during the digestion of carbohydrates. Limit dextrans are the ones on which no further action of the enzyme is observed. And limit dextrans can only be hydrolyzed by limit dextrinase. So only limit dextrinase can break it. Right? So these are the dextrans. Then another important one are the dextrans. Most of the students get confused between dextrins and dextrans. Always remember, dextrins are the bigger units of maltose, but if we talk about dextrans mein there is a mesh of glucose. And if we talk about the linkages, linkage can be alpha 1,4, it can be alpha 1,3. It's like a mesh like this. But it is always formed of glucose. And the most important clinical correlation or clinical importance of dextrans is that these dextrans are plasma expanders. Very, very important. Okay. So plasma expanders, so obviously in case of any injury, you know what happens is infusion of dextrans is given, so it is going to expand the plasma. Then comes the fifth one, or you can say they are also involved in dental plaque. And then comes the last one, cellulose. Now cellulose is a very, very important insoluble fiber. Nowadays, whenever we talk about the concept of diet, because so many people are, switching over to different diets, and there are different implications of different diets. If we talk about carbohydrate rich diet, if we talk about keto free diet, if we talk about protein diet, so depending upon the status, depending upon the condition of the individual, one can switch over to different diets. Okay. So cellulose is generally considered to be ideal one. Why? Because cellulose, it is an insoluble fiber. Fibers should always be included in the diet. And if we talk about fibers, these fibers are actually of two types. One category is insoluble. It is clear from the word itself. and second category is soluble fibers, which are solubilized. So if we talk about insoluble, there are three insoluble fibers: cellulose, hemicellulose and lectins. And if we talk about soluble fibers में gums आते हैं, pectin is there and mucilage is there. And if we talk about gum, the most important example of gums are fenugreek seeds. In Hindi we call them methi seeds. So if we talk about the soluble fibers, be it gums, be it pectins, be it mucilage, the most important function is they decreases the postprandial level of glucose. They always decreases, means they try to bring the level of glucose down. So I can say they decrease the PP or postprandial level of glucose. Insoluble hai, insoluble has to be also added in the diet. What is the basic funda behind that? Because if we are having insoluble fibers, they cannot be digested in the body. Why? Because our body lacks this group of enzymes which can digest them. So cellulose cannot be digested. Can't be digested. And why it can't be digested? Because we lack cellulase enzyme. So obviously if the enzyme to degrade something is absent, there has to be a funda, there has to be some kind of logic why we have to take fibers in the diet. So if it is not being digested, it is going to increase the bowel movements, it is going to increase the peristaltic movements, thereby protecting or preventing our body from constipation. And that is what is the use of laxatives also. Okay. So cellulose ka the main function of this is, or in fact the main functions of all the fibers which we are taking in the diet, the first function is they increase the bowel movements. Very important. Since they can increase the bowel movements, they can actually sequester the bile salts. So if they can sequester the bile salts, they can lead to decrease in the concentration of cholesterol. Third is, just now I told you, they bring the level of glucose also down. So bring the glucose level also down. And fourth is they can be used as laxatives also, and they relieves us from constipation. So these are the four very important features of the fibers. And if we talk about the structure of cellulose, because here we are studying the chemistry, cellulose is linear structure. Very important. It is linear. And the linkage here is beta-1,4. On the contrary, if we talk about the other glucosans, you have seen that the linkage is alpha-1,4. Cellulose only has beta-1,4 linkage, and it is strictly linear in structure. So these are what? These are the glucosans. Then second is fructosans. And fructosans mein again the polymers of fructose, and the only example is inulins in it. Inulins, which are the polymers of fructose. And what is the function of inulins? There is only one function of inulins, that it helps in determination of GFR. Very important. Glomerular filtration rate. And the name of the test is inulin clearance test. Nowadays it has become obsolete, clearance test, because nowadays there are other sensitive tests, creatinine clearance test for RFTs. So inulin clearance test for renal function tests. So these are fructosans. And there is one simile. Most of the students get confused between inulins and insulin. So here I'll just write there are so many similes in biochemistry. Inulin and insulin. The major difference is inulins are carbs, whereas insulin is a peptide, or you can say polypeptide or a protein. Okay. Inulins have fructose, and insulin itself is a hormone which plays a very important role in carbohydrate metabolism. Whereas inulins are carbs which only helps in determination of glomerular filtration rate. So this is all about the homopolysaccharides, in which we discussed the glucosans and the fructosans. Then after this, we come over to the second important class, heteropolysaccharides. Now, hetero means non-identical. Hetero means non-identical, obviously. Now, if we talk about these, we can simply say that these heteropolysaccharides are further of two types, or you can say they have two components. One is they have amino sugars, and second is they have uronic acids. Now, if we talk about amino sugars, simply the most important one is glucosamine. Another one is galactosamine. And if we talk about uronic acid, glucouronic acid and idouronic acid. Uronic acid I have already discussed, glucouronic acid which is produced by uronic acid pathway. And if we talk, these are the components, but they can have sulfate, so I can say they can be sulfate containing. They can be without sulfate also. Again sulfate hai, so charge on them like this. Then they can be acetylated also, so it will be N-acetyl glucosamine or N-acetyl galactosamine. So there are, you can say, different versions of these heteropolysaccharides. Now the question is, you can see they have glucosamine, they have galactosamine. So overall if we see and since it is a polysaccharide, so I can simply say that N-acetyl glucosamine, which is linked to say glucouronic acid, which is further linked to N-acetyl glucosamine. They have polypeptide chains like this. Now, if we have to just summarize them, can I say that they can also be known as GAGs? In fact, they are known as GAGs. Why? Because glycosaminoglycans. So I can simply say that the other name of heteropolysaccharides is GAGs, and this is the most common name. We refer these heteropolysaccharides as glycosaminoglycans, and they are also known as, now where they are present, they are basically present in the synovial fluid. Synovial fluid in the joints, they were first isolated from the cornea. So due to their occurrence and presence, they are also known as mucopolysaccharides. Very, very important. Means since they were first isolated from the cornea, present in the cartilage, present in the connective tissue, present in the synovial fluid, because of that, or you can say connective tissue. So they are mucopolysaccharides. So I can simply say that HPS, heteropolysaccharides, also known as mucopolysaccharides, also known as GAGs. So these are the three terminologies, these are the synonyms. Now the question is, we have studied this, what are the different type of GAGs present? And they are clinically very, very important, because whatever is synthesized in the body, just keep one thing in mind, it has to be degraded also from the body. Nothing retains behind. If something is retained behind, it will lead to a disease. So mucopolysaccharides are synthesized also, they are degraded also. So if we talk about, now I will just refer it as MPS. Now the question is where these mucopolysaccharides are synthesized. So if we talk about the synthesis takes place in the endoplasmic reticulum, and Golgi apparatus. They are synthesized in the ER and the Golgi apparatus, but degradation always takes place in the lysosomes. Now this you have to again remember, synthesis takes place in the ER and Golgi apparatus, but degradation or breakdown is in the lysosomes. And the group of enzymes is hydrolases. So I'll just write in the lysosomes by hydrolases enzymes. So if hydrolases enzyme is absent, there will be accumulation of mucopolysaccharide, which will lead to mucopolysaccharidosis. So I will just write absence of the enzyme will lead to increased concentration of mucopolysaccharide, leading to mucopolysaccharidosis, which is again a very, very important class of lysosomal storage disorders, which we will be discussing after going through the general properties of mucopolysaccharides, right? So now the question is, this is, so we can say synthesis, breakdown, and this. Now what happens is, what are the different properties here, what are the different types of mucopolysaccharides? So if we talk about, I'll just make a table here. So the first one is hyaluronic acid. Right. So if we talk about hyaluronic acid, what are the components of this? So hyaluronic acid consists of N-acetyl glucosamine with glucouronic acid. Right. Second is chondroitin sulfate. Now this chondroitin sulfate is the most common mucopolysaccharide, and if we talk about the composition, it has N-acetyl galactosamine, which is further linked to glucouronic acid. Glucosamine, I'll just write it like this. Right. Then third one is keratin sulfate. Most of the students get confused between keratin and keratin. This is keratin sulfate. And if we talk about keratin sulfate, it is the only mucopolysaccharide which does not have a uronic acid. So here simply N-acetyl glucosamine is there, which is linked to galactose. Very, very important. Just highlight it, galactose means no uronic acid. So no uronic acid is there. And then fourth one, a very important one, heparin. And we all know heparin is the most important natural anticoagulant, right? Heparin, I'll just write natural anticoagulant, and it is the only intracellular gag anticoagulant. And it consists of, again, glucosamine, which is linked to idouronic acid. Okay, this is again a unique feature of heparin sulfate. And again, along with heparin, there is one more mucopolysaccharide which is known as heparin sulfate. So, there has to be a difference because the individual components are different for every mucopolysaccharide. So, if we talk about heparin sulfate, don't get confused between heparin and heparin. Heparin sulfate, there is a difference in both. This consists of glucouronic acid instead of idouronic acid. So, the components are glucosamine plus glucouronic acid. And the last one is dermatan sulfate. And dermatan sulfate has N-acetyl galactosamine plus idouronic acid. So, these are, this is the list of the mucopolysaccharides. So, I will just write the name here and these are the components. Okay. So, I can say hyaluronic acid has N-acetyl glucosamine, glucouronic acid, chondroitin sulfate, which is considered to be the most common mucopolysaccharide, which has N-acetyl galactosamine, glucouronic acid. Keratan sulfate has N-acetyl glucosamine and galactose. No uronic acid is there. Heparin has glucosamine and idouronic acid. Heparin sulfate has glucosamine and glucouronic acid. So, this is one of the differences also between heparin and heparin sulfate. So, these are the important ones. Now, if we talk about the functions, hyaluronic acid plays a very, very important role in, I would say, morphogenesis, in metastasis, then you can say in so many, it helps in recognition also in the sperms during the process of fertilization because hyaluronidase is the name of the enzyme. So, this is a very important, I would say, mucopolysaccharide which is actually, it plays a role in inflammation also. So, there are numerous roles of hyaluronic acid. But the most important point which you have to remember is hyaluronic acid lacks sulfate. Or this is the only mucopolysaccharide which does not have sulfate in it. Sulfate hai to charge hai. Similarly, keratan sulfate is the only mucopolysaccharide which lacks uronic acid. And dermatan sulfate or chondroitin sulfate, they are the most common found mucopolysaccharides or heteropolysaccharides because dermatan present in the skin, it is clear from the word itself, and chondroitin sulfate is further of 4 types A, B, C and D. Basically, dermatan sulfate is nothing but chondroitin sulfate B. Now, the most important point which we have to remember here is, which we are going to discuss in detail, one is keratan sulfate. Now, keep one thing in mind, the most important function of this keratan sulfate is, just now I told you it lacks uronic acid. And this keratan sulfate is actually, this was the first mucopolysaccharide to be isolated. To be isolated from the cornea. And this keratan sulfate is of 2 types, keratan sulfate 1 and keratan sulfate 2. And the basic difference between 1 and 2 is that keratan sulfate 1 is present in the cornea and keratan sulfate 2 is present in the loose connective tissue. Loose connective tissue. So this is one very important point which we have to remember about keratin sulfate. Then second is heparin. Now if we talk about heparin, we know what are the important properties of heparin. The most important one is it is the only intracellular gag. Intracellular gag. This is one very important property. Second important property of heparin is that we know that it has, just now you saw the components, it has isouronic acid, and this is the most important anticoagulant. Which binds to T-thrombin 3. Thrombin 3 factor. And the third important function is it dislodges lipoprotein lipase from the surface. So it dislodges lipoprotein lipase from the anchoring surface. Then coming over to the one parallel to this, heparan sulfate. Heparan sulfate. Now if we talk about heparan sulfate, this is a very, very important mucopolysaccharide. Why? Because it is lining the basement membrane in the kidneys. So it lines the basement membrane in the kidneys. Okay, so in this bargain what it is doing, since it is lining, so it will prevent albumin to pass through it because it is negatively charged. So it will prevent the excretion of albumin, which is very, very important because we know if albumin starts coming in urine, microalbuminuria, which is the most sensitive indicator of DM. So it prevents the excretion of albumin being charged or being negatively charged, whatever you can say. Charged. And the third important function is, जैसे it is dislodging, it is helping lipoprotein lipase. So it helps lipoprotein lipase on the endothelial surface. So this is the basic difference and the functions of heparan sulfate and heparin. And the last one, if we talk about, it is dermatan sulfate. Now dermatan sulfate mein the most important point which we have to remember is that this dermatan sulfate, it is a atherogenic gag. Atherogenic gag, the most important one. So it will basically help in transport of LDL, and we all know that LDL transportation is, LDL is known as a bad cholesterol because it helps in taking cholesterol from the liver and depositing it to extrahepatic tissues. That is why it is atherogenic gag. So helps LDL in a transport of cholesterol. Or you can say transport of bad cholesterol, we can say. So these are the four very important mucopolysaccharides. Hyaluronic acid is also there. Just now I told you it helps in, I'll just write about hyaluronic acid also. Main is, what is the function of hyaluronic acid? It is required for wound healing, in metastasis, morphogenesis, embryogenesis, and this is the only mucopolysaccharide without sulfate. So this is also very important. So hyaluronic acid is the only mucopolysaccharide without sulfate, and keratin sulfate is the only mucopolysaccharide which lacks uronic acid. So these are the general properties of all the mucopolysaccharides. Now coming over to what is the clinical correlation of this. So whenever we talk about the clinical correlation, it leads to mucopolysaccharidosis. And how do we define these mucopolysaccharidoses? They are simply the lysosomal storage disorder, disorders in the lysosomes due to the deficiency of hydrolysis group of enzymes, group or class of enzymes. Now, what are the different type of mucopolysaccharidosis? And if we see the diagnosis, it is simply excretion of GAGs in urine. So if we have to diagnose a patient suffering from mucopolysaccharidosis, there will be excretion of these glycosaminoglycans in urine. So now first of all, what are the main clinical features of mucopolysaccharidosis? So if you see, there are most commonly features seen in all different type of mucopolysaccharidosis, but there are some 1 to 2 features which can distinguish between one mucopolysaccharidosis to another. So normal is frontal bossing will be there. Then depressed nasal bridge. Right, there will be pain in the groin area. And it will lead to different type of hernias, whether it is umbilical hernia or it is inguinal hernia. Then another important feature is copious nasal discharge. All the time there will be a discharge from the nose. Right, and there will be a defect in the vision, so corneal clouding will also be there. And if we carry out the X-ray of that, so radiographic survey will be what? It will lead to breaking of the vertebrae. I'll just show you the pictures also. Breaking of vertebrae. Right? There will be a dystosis complex. And due to degeneration of the metacarpals, it will lead to bullet-shaped phalanx. Right, and just show you the images also. So if you can see these images, right, this is the depressed nasal bridge. If you can see this, depressed nasal bridge is there, frontal bossing is there, short stature, intellectual disability, obviously mentally retarded. Then leukocyte inclusions, this is also very important. These are known as Rayleigh bodies. So Rayleigh bodies will also be there. Then coarse facial features, as you can see, joint contractures and dysostosis complex. This is very important, dysostosis. And if we talk about, and hepatosplenomegaly will be there because enlargement will be there. And if you see the X-ray of this, you can see the X-ray also. This is the degeneration of the metacarpals. And there will be breaking of the vertebrae also. And this also is a picture where you can see the umbilical hernia is there. And if you clearly see, you can see that the vertebrae are also like disfigured. So this is actually mucopolysaccharidosis, where the basic features are corneal clouding, Rayleigh bodies, short stature, intellectual disability, coarse facial features. But the only thing is that there are different type of mucopolysaccharidosis, how to differentiate between them. So what are the different type of mucopolysaccharidosis? I will just highlight it here. Okay, this is the breaking of the vertebrae. Now, the different type of mucopolysaccharides mein, the first one is MPS 1H. This stands for Hurler's syndrome. Then MPS 1S. This stands for ski disease. Then MPS 2 stands for Hunter's. Then MPS 3 stands for Sanfilippo's. MPS 4, there is no MPS 5, and MPS 6. So if we talk about MPS 4, this is Marquis syndrome, and 6 is Marrow-Twix-Lamy syndrome. Twix-Lamy syndrome. Now one more has been there, MPS 9, which is Nato-Wicks syndrome, N-A-T-O, Nato-Wicks syndrome. So these are the different type of mucopolysaccharidosis. Now if we talk about the deficient enzymes here, first of all we will take the deficient enzymes. In Hurler's syndrome, the deficient enzyme is alpha-L-iodouronidase. In ski syndrome also, the enzyme is the same, alpha-L-iodouronidase. But the only thing is that it is partially defective or partial defect. In Hunter's syndrome, the deficient enzyme is iodouronate sulfatase. In Sanfilippo syndrome, the enzyme is similar to that of heparin sulfate. So enzyme same as heparin sulfate. If we talk about Marquis syndrome, in Marquis syndrome, the deficient enzyme is galactosamine 6-sulfatase beta galactosidase. And here in Marrow-Twix-Lamy syndrome, it is N-acetyl galactosamine 4-sulfatase. Samine 4-sulfatase. And if we talk about Nato-Wicks, it is hyaluronidase. So these are the deficient enzymes in mucopolysaccharidosis. This is the name of the disease. Now the question is, all of them are going to have more or less the similar features, but what are the different features by which we can distinguish these different type of mucopolysaccharidoses? So if I talk about simple, I will just write it here, the classification as Hurlers, Ski, Hunters, Sanfilippo, Marquis and Marotwick-Lamy syndrome. Right. First of all, the most important point which we have to remember here is that all the mucopolysaccharidoses are autosomal recessive except Hunters. So, if we talk about the mode of inheritance, all are autosomal recessive, only this is X-linked. That means Hunters is there present only in the males, and this is autosomal recessive, autosomal recessive, autosomal recessive. Then another point of differentiation is that one is mental disability and corneal clouding. So, if we talk about Hurler's syndrome, Hurler syndrome में both are present. In Ski syndrome, because you saw that in Ski syndrome the name of the enzymes were same, it is alpha-ylidouronidase, but Ski disease में it is due to the partial defect of alpha-ylidouronidase. So mental disability will be absent. Absent in the sense that Ski में the baby who is born with the Ski disease will have a normal intelligence. But the most important property here will be he will suffer from hirsutism. That I will write later. And if we talk about corneal clouding, it will be present. Then coming to Hunters and Sanfilippo, always remember one thing. In Hunters, intellectual disability will be there. In Sanfilippo also it will be there. But corneal clouding will be absent. Absent here. Okay, for this I will tell you one mnemonic also to remember. Now coming over to Marquis. Marquis mein it is absent, but corneal clouding is present. Here it is absent, but corneal clouding is present. So this is how we have to differentiate between different type of diseases. यहाँ पे I can just write inheritance and here the disease. So the most important one is Hunters is X-linked, right? And corneal clouding is absent. For this we have to just remember one mnemonic. Sam goes for hunting. The mnemonic is Sam goes for hunting. And it is very well clear, if we can't see, we cannot go for hunting. So to hunt something, to kill something, one has to need to have a clear vision. So in Sanfilippo and in Hunters, the vision is clear. So Sanfilippo and Hunters, the vision is clear. No corneal clouding is there. This is one very important point which you have to remember. Second is, if we talk about Ski disease, always remember in Ski disease, obviously the normal intelligence will be there. The child will be born with a normal intelligence. But the most important property here will be, the feature will be hirsutism. The female who is born with Ski syndrome will have hairy body, या hair all over the body. Okay, so these are the two very important points. And third important point is, all mucopolysaccharidoses are autosomal recessive except Hunters, which is X-linked. That means it is present only in males. Only in males. So these are the three important points, and the last point is that Rayleigh bodies are present in all except Marquis syndrome. Rayleigh bodies, yes, right, Rayleigh bodies present in all mucopolysaccharidoses except type 4 Marquis syndrome. Rest will be similar in all the cases. So these are certain exceptions which you have to remember. Rest, the features along with diagrams I have already explained. Then we come over to the most important one. What is the treatment of this? So if we talk about the treatment, one is stem cell therapy is there. And if we talk about the stem cell therapy, it is basically available for mucopolysaccharidosis 1H, 1S. H stands for Hurler's, S stands for Ski syndrome, and for Maroteaux-Lamy syndrome also MPS 6. And if we talk about the second one, enzyme replacement therapy, which is the most commonly used therapy, enzyme replacement therapy. And this is available for 1, 2 and 6. And what are the enzymes? We can simply say that L-durazyme, elaprase and Nagella enzyme. So these are the two main therapies which are available to treat different type of mucopolysaccharidosis. Stem cell therapy is required for MPS 1H, MPS 1S and MPS 6, and enzyme replacement is for 1, type 2 and type 6. Because nowadays recombinant DNA technology is also rising, and it is also required for treating, because what we can do is we can recombine DNA if it is a link like that. Okay, so this is all about mucopolysaccharidosis and mucopolysaccharides. And here in this video we finish up with the chemistry of the carbohydrates. And in the next video we will be discussing about the different transporters of glucose, and then we will discuss with the metabolism. So I hope this video is clear to all of you. Thank you so much.